I had EFM in aerospace engineering in 2003 under the advising of Alex Gordetsky and remained in the lab for an additional year as a postdoc. In the Gordetsky lab, Nick researched how to use stochastic models of dynamical systems to improve system identification. Now, Nick works in the Rajapakse lab researching how to create data-driven models of the dynamics of cellular programming. So without further ado, Nick, please take it away. Thank you.
All right, yeah, thank you, Sahil, for that nice introduction, and thank you, Sahil and Aniket, for the invitation to talk. Very excited to be here. Right, like Sahil said, I got my PhD in aerospace, and then I really want to take my computational skill set that I had acquired during my PhD and bring that to a more scientific discipline. So first, I'll begin by introducing some of the collaborators that also have worked on this research with me. So the PI for this project is Indika Rajapakse. Also working with my former advisor under this project, Alex Gordetsky. We also have another professor in the lab, Lindsay Muir, and lab manager, Walter Meixner. And we also have a lot of great PhD students in the lab, Cooper Stansberry, Joshua Picard, Jillian Quishaishan, and Mark Che from the mathematics department. So these people have all been really great in helping me learn the ropes of biology and getting started on the project. So I'm going to start just by giving the main motivation. Sorry, let me hide these controls first for this work. So for those of you that are familiar, when a person is diagnosed with cancer and they undergo treatment, they have to go through chemotherapy. And in chemotherapy, this can be very hard on the patient because it really destroys the immune system and is very difficult. And so one of the ways that they will replenish the immune system is through a bone marrow transplant, where they will take bone marrow cells from a donor and then give them to the patient. And this is meant to replenish a lot of the immune cells in the body. But the issue with this is that there's something known as graft versus host disease, GVHD, where since the donor cells are not produced by the patient's immune system does not recognize them and will try to attack them. And so one of the solutions for this could be if you were to take the patient's own cells and reprogram them into immune cells and use those to rebuild the immune system. In this way, you wouldn't have this issue where the cells are foreign and then you have your own immune system attacking these donor cells. So this is really the main motivation behind the work of this lab, is trying to develop this technology. So even broader than that, what if we could take, for example, a skin soft person and reprogram it into any other cell, like a cardiomyocyte, red blood cell, and so on. So there's many applications to the cell reprogramming. One, like I mentioned first, is replenishing the immune system. Another one could be tissue regeneration, where if you have tissue damage from some sort of disease, you could replenish it using your own body cells by the cellular reprogramming. Drug discovery, for example, taking cells and reprogramming them to a certain type of cell that you're interested in and testing drugs on those cells. And also disease modeling, if you can program a cell into a disease cell and then study how that cell behaves. So there's a number of challenges that still remain that we want to tackle. Time. So if you're doing experiments where you're trying to decide or trying to figure out what is the best way to reprogram a cell, and I'll go into more details later on about how these experiments are conducted, but it can take several weeks to grow the cells, perform the reprogramming experiment, and wait for the reprogramming to occur. And the other one is money. So one experiment is going to take at least $15,000 as well. So you're very limited by how many experiments you can actually do. And so the idea is, can we do all these experiments in silico, computationally to identify what might be worthwhile experiments to do and drastically accelerate the discovery of these cell reprogramming methodologies. So in this talk, what I'm going to do is first give a description of how genetic information flows throughout the cell, and then I'll discuss cell reprogramming and the mechanics of it. And then I'm going to introduce this concept of digital biology. After that, I'll go into one of the foundation models that we're going to be using for these in silico experiments and describe its architecture and structure. And then if we have time, I'll also discuss some other work that we're doing on cell reprogram where we try to learn cell cycle dynamics. And then lastly, I'll finish with conclusions. So first, flow of genetic information. Biology is largely an information system where all the information is coded in the DNA and it is transported throughout the body to perform a lot of different cellular functions. So if we look at the human genome, where all this information is stored, at a high level, we have the cell and all the DNA is stored within this nucleus. So in the human nucleus, we have 46 chromosomes, and here's an actual picture of them using a technique where you can actually paint each of the chromosomes. So it's pretty cool to see them. The genome, what it is, it's long strands of DNA. So I have a little bit of a schematic here where this black line, it can represent the DNA. And then you have these regions of the DNA that are known as genes. And if you look at how much of the genome is actually genes, it's actually a very small number, but this is where a lot of the information for cellular function is stored, is in these genes. So the main flow of this work is, or of cellular function, is that the DNA makes RNA and then the RNA makes protein. And the protein is what is responsible for nearly all cellular functions. So which RNA is made is going to determine which protein is made, and then which sort of functions the cell is going to perform. And we have a schematic here showing that if you have a strand of DNA, you have these segments known as genes. The genes will go through transcription, which produces the mRNA. And then the mRNA is converted into proteins. And whichever proteins are in a cell is largely representative of what sort of cell type they are and what their function is. And the human genome contains around 20,000 genes, so it's a huge number in a very high dimensional system. So now, how is this system regulated? There's something known as transcription factors. So these are specific proteins within the cell that are responsible for turning on or off certain genes. So for example, we have 3 different types of transcription factor, or of genes here. Also G3. So this is where a gene produces RNA, which is then made into a protein, and that protein will leave the nucleus and then go out and perform different cellular functions. Then inside of this box, we have transcription factors. So a gene produces RNA, which is produced the protein, but then this protein can go back to other genes and influence their transcription, influence how much of the RNA is produced from these other genes. And then inside of these transcription factors, there's also something known as master regulators, where the proteins can then go back to the gene that first transcribed the RNA and regulate it as well. So this is going to be the main mechanism through which we're able to control the transcription of RNA within the cell. And then something else that's important is how chromatin, which is basically like the structure of the DNA or of the chromosome, is packed within the nucleus. So you can think of it as like, spaghetti noodles packed inside of this little tight ball. And what researchers have found is that there are these clusters known as transcription factors, where you can have genes from different chromosomes that will all interact to produce certain biological functions within the cell. So you can have these dependencies within the cell that are very far apart if you're just looking at the one-dimensional genome. And so it's important when you're looking at function is where do these different genes interact, and trying to identify the long-range dependencies within the transcription. So now I'll go into a description of cell reprogramming and what we're trying to do. So in typical cell development, there's something known as the Waddington's landscape. This was an analogy that was developed by Waddington to describe how cells normally develop and how it's like a tightly regulated process where all cells have can trace their lineage back to an embryonic stem cell. And then as the organism develops, these stem cells can differentiate into different types of cells, all performing different functions. So from an embryonic stem cell, you will get every other type of cell type in the human body, which is about 300. So researchers initially thought that this was like a one-directional process where all
Can you
cells
hear me?
start with an ancestor stem cell and then eventually differentiate fully into one of the final human cells. But what we found is we had a researcher by the name of Harold Weintraub, who in 1989 was the first one to show direct reprogramming. I think we have a question in the chat.
Can you hear me?
Yeah, we can hear you.
Yeah. So, so how does so are you saying you have a stem cell and then that transforms into other cells? Is that what you're trying to say here? Or these are all different cells and they're...
Yeah, so I'm saying that all cells can be derived from an embryonic stem cell. So as the stem cell develops, different functions will turn on and it'll follow certain paths, different lineages to other cells.
Okay, but everything comes from the stem cell. Is that what you're saying?
Right, yeah.
Okay, all right, thanks.
Good question. Right, so Harold Weintraub in 1989 showed that we can do direct reprogramming. So what he did, he took a fibroblast cell and added this transcription factor MyoD to it and showed that that would transform it from a fibroblast, which is like some sort of like connective tissue cell, into a myoblast cell. So it's like a skeletal muscle cell. And so this showed that this process, this differentiation could actually be manipulated by external variables such as these transcription factors. And then another researcher by the name of Shinya Yamanaka showed that we could actually go back up the landscape. And he showed that with these 4 transcription factors, you can take a fibroblast cell and go to an induced pluripotent stem cell. Basically like an embryonic stem cell that's come from this reprogrammed cell. And so now we want to be able to find out different recipes that can transform a fibroblast cell to other types of cells instead of just a pluripotent stem cell or a myoblast cell. And the way that we're going to do this is by controlling the flow of information. So here I have a schematic of a cell with the nucleus. And if you remember, we have the genes inside the nucleus, which make the mRNA, which then go on to make the protein. What we can do is add in modified mRNA. So this would be like a transcription factor into the cell, and that will stimulate the production of RNA from a specific gene that this transcription factor targets. The other thing we can do is suppress genes. So this is known as RNA inference or interference, where we can add in a transcription factor that will turn off a gene. And this allows us to manipulate this information network within the cell. So something important to note here is that each one of these modified mRNA transcription factors costs $15,000. So each experiment that we do, depending on how many transcription factors that we use, can be very expensive. And another thing is that the number of genes in the genome is around 20,000. Transcription factors is 1,800. So just trying to find out which transcription factors can perform the reprogram that we want can be an immensely expensive task if we were to just try to brute force it through like combinatorial search. So the idea is that we would like to be able to simulate this flow of information on a computer so that we can really narrow down the candidate transcription factors that would be useful in reprogramming a cell. So this is now where digital biology comes in. So let me first introduce the concept of digital biology. I think this quote from the founder and CEO, Jensen Huang of NVIDIA, does a good job of stressing the potential of it. He says, where do I think the next amazing revolution is going to come? And this is going to be flat out one of the biggest ones ever. There's no question that digital biology is going to be it. So what is digital biology? Well, digital biology is this recent field that's been created as a result of the advancements on a lot of technologies in other different fields. So one thing would be the exponential increase in data availability. We now have more biology data than ever before. There's been an exponential increase in how much data we have. And to get a sense for this, we can go back to 1999, where there was the Human Genome Project. It took them around 13 years and $3 billion to sequence the human genome. And today we can do that in just one day for about $600. So now it's much easier to sequence DNA, which brings me to the next point, which is that we can now read, write, and even edit DNA using technologies like CRISPR. And then also there's technology that allows for automation of the lab, so that these experiments can be run 24/7 and be run in a very precise and repeatable way. What I have down here, this is a picture of a robot that we have in the Rajpaksi lab, known as the BioAssemblyBot or BAB. And I included a video of how it works because I think it looks awesome. Let's see. So in this video, you can see it taking a plate of samples. Oh, not on Zoom. Okay, let me. No, let's just do screen share, general screen share. So it's a robotic arm, and there's different what should I like, not like heads, but like extensions, I guess, that you can attach to it for different purposes, for like pipetting. But here it's moving a plate of cells out from this, I don't know what to call it, like a hood, into some sort of measurement apparatus. Here. And then another important advancement that's enabled by digital biology is deep learning and AI. As we've all probably heard recently, there's AlphaFold, which recently won the Nobel Prize. And it can predict foldings of different proteins. And it's even been able to predict novel proteins that have not been discovered yet. And then we've even been able to experimentally validate the I guess existence or the validity of these novel proteins that were discovered with this neural network. This also allows for digital laboratories. So what we're working on in the Rajpaksi lab, for instance, is basically a digital assistant for biology, or almost like a digital twin of the lab, where we should be able to like make experiments or software to run experiments, have the robot run it, take the data, have it be processed automatically, and use that to update our models, and use those models then to create or inform another experiment, and have this loop between the real lab and the digital lab. This digital assistant is a paper that has recently put on archive from the lab, where it's a large language model with a RAG, a retrieval assisted or augmented generation, where you can add in different documents, and that can help tailor the response of the large language model towards being more knowledgeable about biology based on the documents that you supply it. And then the other thing here that we have a recent paper, automated wound generation. So these pictures show microscope images of wounds that were created within skin cells created by the robot, the BAB. And you can see it has very high precision that would not be possible just with a human making these wounds. So it's very exciting that we're able to have the robot and also develop these technologies that can eventually get us to a place where a lot of this can be automated. And now I want to give a sense of the different data that is available for biology and that we're going to be working with. So first off, this is going to be one of the main types of data that we work with for this presentation is single cell RNA sequencing. So the way this works is you have a collection of cells that you put through this box. It's very small. It's around like 6 inches in length. And what it does is it will, what's known as lyse the cell, basically break the membrane and take all of the RNA molecules out of the cell and then count each of the different types of RNA molecules. And it gives you this sparse counts matrix as an output. And the dimension is going to be cells by genes. And there's around, 20,000 human genes. And if you put it into this Prometheon, you can expect some technical to drop out, and you end up usually around with about 15,000 genes. Another type of data is chromatin conformation data. So if you remember how I talked about the transcription clusters or the transcription factories, where you have genes from different chromosomes that end up close to each other and producing different RNA molecules. This is what the HiC is capturing. So it will look at how close two different regions of the genome are to each other. So each one of these green lines here divide the matrix into different chromosomes, and it can show how much contact there is between different regions in those chromosomes. And the size of this is going to depend on the resolution. So here, down here, we have 1 megabase. So the base is like one pair of nucleotides. Would be a 3000 by 3000 matrix. But if you have a very fine resolution of 50 base pairs, then the matrix becomes extremely large, 62 million by 62 million. So the data that we work with can be extremely high dimensional and be very well suited for machine learning. And then another type would be chromatin accessibility. So the structure of the chromatin can determine which regions are available for gene transcription and then which ones are closed off. And the way that's determined is by how tightly coiled the chromatin is. So if it's tightly coiled, there's not as much space for things to bind onto the DNA and to produce transcription. So that's what's known as heterochromatin. And then if it's very open, then there's more space for things to bind and they can produce the RNA molecules, and that's known as euchromatin. So we can also measure this and get a 1D vector corresponding to the length of the genome that shows where things are heterochromatin and where the chromatin is euchromatin. And lastly, we also can do videos. So here we have, we can add what's known as a reporter into cells, where if you put it under UV light, it will shine, it will glow different colors depending on the reporter and what that reporter is targeting. So we have a green one here. So let me back up. At the bottom, we have different phases of the cell cycle. So the cell cycle is how the cells can proliferate, split into two. And you can see that there's different proteins that are involved in different phases of the cell cycle. And then these reporters will glow when there's more presence of these different proteins. So if we look at cells only, we can see that they're going through the cell cycle. We have some that are more like around the G2 phase that are red, and then some that are closer to the earlier phase, the G1 that are green. And now these are experiments where we add in transcription factors, and we can see how that affects the progress of the cell cycle. So for instance, you add in MyoD1, or you can suppress, for example, PRAX1. You can see there's a lot more activity when we suppress PRAX1. So it's very cool that we can see this type of data just through this live cell imaging. So now let me get into the actual Geneformer or the foundation models that we're going to be using to do the in silico experimentation. So just first a brief introduction of what is a foundation model. At the heart of it, it's a neural network where you've taken a ton of data from a lot of different sources. It can be multimodal, it can be unimodal, and you use that to train the foundation model, the neural network, based on what's known as a pretext task. So for example, masked language modeling. And what this training does is it teaches the foundation model to be able to extract the semantic meaning from data, basically. It's able to pick out very important qualities of the data, and those embeddings that have those embedded qualities of the foundation model can then be used for a lot of different downstream tasks. And so a lot of times, these embeddings can be used to outperform simpler neural networks that have not had the benefit of being trained with a huge amount of data. And what's important about or like the breakthrough technology of this foundation model is the attention mechanism, where the foundation model is able to learn what's known as a contextualized embedding, where each token or word is going to be mapped to a different embedding depending on the value of other tokens in that sequence. So there's like a figure here showing how different words are going to be important for certain other words within the sequence. And we have something similar in the genome where different genes are going to interact with other genes, and it's going to be very dependent on the context of things like cell type and of the expression of other genes. And so conceptually, this mechanism should be very well suited to identifying the long-range interactions of the genes within the genome. And so that's sort of the idea behind trying to take the success of foundation models and applying those to biology. And the issue here is that we don't have sentences in biology. We don't have anything that is sequential like this on the genome. So we need a way to convert genetic expression into something that is amenable to the foundation models that have already been created for natural language. The way that Geneformer does this, Geneformer being the foundation model that was developed for RNA counts for single cell RNA sequencing data, is that it will look at your data. So you remember it's this matrix cells by genes, where for each cell, it will count the number of RNA molecules produced by each gene. And that's what this raw data in the top row is. Geneformer will do a rank value encoding where it reorganizes this ordering so that the most highly expressed genes are at the top of the vector. And then it will truncate it at around 2048 genes, just so that it's still computationally feasible. And then once you have this ordering, you will map each of the genes to a token. And all this is done taking into account like baseline expression of genes. So everything's normalized to make sure that, for example, housekeeping genes are not considered to be highly expressed when they're just these genes that might always be present in cells. So, and what the architecture looks like is you'll take this rank value encoding that I've shown here in red, you put it through the transformer layer, and you get out a gene embedding, which is the size of 2048 by the hidden dimension of 256. So this is basically just the exact same BERT architecture that's been used in a lot of other foundation models, but now the input has been structured in such a way that the genetic expression is amenable to putting it through this existing architecture. And what we get out, what we're specifically interested in is the cell embedding. And so you get this just by averaging across all of the rows, and then you get some sort of 256-dimensional embedding that we hope will be encode all of the information that will be useful for detecting, I guess, whether or not cell reprogramming is successful, or it will encode all of the useful qualities or distinguishing qualities of different cells. So now let me get into the experiment. The first thing you need to do when you use these models is fine tune it. Fine tuning is where you have some sort of downstream task that's tailored to your goals or your experiment of interest that you will update the pre-trained weights of the model on. For this, we use cell type classification. Our goal again is going to be looking at how adding transcription factors changes cell identities. So we want the model to be very good at identifying different cell types. And you can see here, I have shown UMaps of the pre-trained embeddings on the left, and then the fine-tuned embeddings on the right. You can see after fine-tuning on this cell type classification objective, the clustering is now the cell type, whereas before it looks like maybe it was more according to data set. And now afterwards, the data sets are not so separate. You can see there's a lot of overlay between different data sets within these clusters. So this is how we know that the fine-tuning was successful in identifying different clusters of cell types. And the two that we're going to be interested in here are the fibroblast cells and the hematopoietic stem cells, HSC cells. And these are right next to each other, so we feel that they should be good candidates for reprogramming, but also we know from literature that it is possible to reprogram fibroblast cells to HSC cells, so that's another reason why we choose these two specific cell types. Now let me explain how this perturbation experiment works. So again, we start off with this fibroblast rank value encoding, and the idea is in like the lab experiment, what you would do is you would flood the cell with a transcription factor. So what we do in silico is we say, okay, let's say that these transcription factors are now the most expressed genes within the rank value encoding. So we add that to the top of the rank value encoding, and then again, truncate, so it's still 2048, the sequence or the length needed for the architecture, and then feed that through the Geneformer and look to see how the embedding changes or shifts between the unperturbed input and then the perturbed input. And there's a few caveats I want to throw out before getting into the results. So all transcription factors are added to the top. So this doesn't take into account that the transcription factor might not be the most highly expressed gene when you actually do the experiment. It doesn't take into account the order of transcription factors. We're not going to try doing 1, 2, 3 and 2, 1, 3 and so on and so forth, just because that really grows the search space very quickly. And then this only models the first step of the cell state transition. So when you reprogram a cell, you add this transcription factor. That transcription factor is going to influence other genes. Those influenced genes now influence another set of genes, and there's this cascade of gene effects throughout the cell. With this foundation model, you only get really the first layer of how this transcription factor might move the cell. But the relevant measure that we're going to be looking at is the directionality of the shift. Is it pointed in the direction of our target, or is it not? So the experimental setup that we have for this is we have a set of 10 candidate transcription factors. These are a set that we have identified in the lab that might be good for reprogramming, and then some of them are also present in the literature as well, affirming that our estimates are meaningful. And what we'll do is for these 10 transcription factors, we'll choose 5 of them. So, and we'll try every combination of those 5. So what it ends up being is 252 total recipes that we put through the Geneformer. And what we'll look at for the distance metric is the cosine distance. So it's defined as being 1 minus cosine of the angle between 2 vectors to see if they're pointed in the same direction or not. And then we'll normalize it to look at only the relative distance, where we look at cosine distance between the perturbed cells centroid, or the centroid of the perturbed cells, and then the centroid of our target HSC cells. So for this analysis, we're only going to be looking at centroids of the cells. We have about 34,000 fibroblast cells and 20,000 HSC cells. So the results that we get, you can see in the histogram, the negative values are better. That means that they're closer to the target, and the positive values are farther away from the target. I have the red line here at 0 to show where the initial unperturbed cells lie. And it's roughly half and half which cells have gone closer to the target and which have not. And then we have a list showing the recipes that give the best movement towards the target and those that give the worst. And if we look into the literature, we actually find that GATA2 is very important within HSC cells and very important for reprogramming fibroblasts to HSC cells, which is contradictory to these results, indicating that there is still some work to be done bringing foundation models into biology. And we have some ideas about how to improve it now, and some hypotheses of why maybe this is not so reflective of what we know to be true in biology. One of them is the scale. So if you think about the scale of some of the most successful foundation models like GPT-3, you can have upwards of 175 billion parameters and 45 terabytes of unfiltered data. Comparing to this model here, there's only 10 million parameters, so it's very, very tiny. Even like the smallest GPT-3, I think, had around 25 million parameters, and it was only trained on 30 million cells. So one thing that we'd want to do is scale up. There is another Geneformer version that has 106 million parameters, and we did see results that were agreed more closely to what we know from the literature, especially with regards to GATA2 being at the top. So that does give us some sort of hope that it might be a scaling issue. So we would like to compile, a database or an atlas of cells that can be used for training a very large scale Geneformer. And the other thing we want to do is to incorporate different types of data. So I laid out the different types of data that might describe the function or the state of a cell. And we think that maybe gene expression is not enough to fully know what like the context or the function of the cell is. So we'd like to be able to add in these other types of data, especially, for example, the chromatin conformation, the HiC data. This is a matrix, or you can even be thought of as an image. And so one challenge is going to be, how do we incorporate these different modes of data into a larger Geneformer architecture? And there are architectures out there that can work with like text image pairs, but usually those have are paired together. So the text pertains to the image and the image reflects what's in the text. Whereas when we have data in biology, a lot of times they'll come from different cells. They're not exactly paired, or you might just have one or the other type of data. And so that's another challenge, is how do you deal with when you sometimes have both the HiC and the gene expression data, and sometimes you just have one or the other. And another challenge is that if you want to train an architecture with 175 billion parameters, for instance, it's going to be a lot of work to set up that training pipeline, and also a lot of compute is going to be needed to be able to train so many parameters with so much data as well. So this is something though that we want to work with NVIDIA on, and we have been talking a little bit to some representatives from there. And so hopefully this is something in the future that we can start to develop and come up with a foundation model for single cells that will be able to enable much better like machine learning methods or architectures in the future using the embeddings from the foundation model. So how much time do I have? Very briefly, I'll also describe another body of work that we're doing for cell reprogramming, which is trying to learn cell cycle dynamics. So even when we know a good recipe for reprogramming, a lot of times the efficiency of reprogramming is only around 5% or even less of cells will actually be reprogrammed. And one idea we have for why this is the case is that it depends on where the cell is in the cell cycle, and how transcription factors will affect the cell differently at different phases or different time points within a phase of the cell cycle. So what we'd like to do is learn the dynamics of the cell cycle. So just a brief overview of the cell cycle. And let me also back up and say that the reason why we're interested in the cell cycle is that cells that are going through the cell cycle, cells that are cycling, oftentimes have more of their chromatin opened up. There's more gene expression, and it's easier to manipulate when they're cycling through the cell cycle. There's 4 different phases within the cell cycle. There's G1. This is the longest phase at the beginning. And then the next phase is S. So this is where the second genome is synthesized. And then G2, G being gap. So another gap where it prepares for the next phase, which is mitosis. This is where the cell splits into 2 daughter cells. And the problem that we have about a system identification setup, the data that you have will be trajectories. You'll be able to measure one object over an extended period of time. But here, if you remember, when I talked about single cell RNA sequencing, you would take cells at one point in time and then kill them in order to measure the RNA molecules in the cell. So you cannot measure a given cell over a period of time. You can only measure at one time point. So what we have is, the way we do this, we have a collection of cells. Each one is going to be at a different point in the cell cycle, and then we measure them out once. So now, theoretically, we should have cells that are densely sampled over the course of the cell cycle. And of course, it's not the same cell, but it should be representative of how the transcriptional profile should look for a cell throughout the cell cycle. And then we need to be able to order where each of these cells falls along the cell cycle. So the way that we do this is there's a method known as pseudotime, basically where it assumes that the biological process, in this case, the cell cycle, is continuous, and then it will look at the graph to come up with a distance between 2 cells. And the way that it does this is that we use a Markov process, basically a random walk, and look at what is the probability of going from one cell to another cell, and then using that probability as a distance. And you can normalize that probability and then put everything in between 0 and 1, and you get this ordering of cells. The only thing you need is to specify the root cell. And then you can say, okay, if there's like a lot of connections leading to the cell, or like a very short number of jumps to get to the cell, then maybe it's nearby. So for example, this is different than just like a Euclidean distance, where if you look at this grouping of cells here, these blue cells might appear very close to the yellow cells in Euclidean space, but actually they're very far apart if you look at the trajectory of the process. It's about over halfway through, whereas these are from the beginning. So this pseudo-time method is meant to disentangle like the Euclidean distance from actual the progress throughout the process. And to do this, of course, since it does assume that it's a continuous process, we do impute the missing values so that things are much smoother. Let me see. So the data that we have from this, I've sort of already mentioned this. First, it's a grouping of cells, and we're able to also measure the phase or an estimate of the phase of each of these cells, G1, S, and G2, using a fluorescent marker. So we'll have a collection of cells, and then we order them by pseudotime. And then we also have two transition points that we can measure. So there are different ways to suspend cells throughout the cell cycle using different blockers and inhibitors. So this allows us to get a profile of the root cell, MG1, and then we're able to use that for our pseudotime algorithm. And we come up with a pseudotime ordering of the cells. So here in the top, this is the data that we have in the cells projected onto the UMAP plot. You can see that the G1 is blue. This is the beginning of the cell cycle, which most of it is contained in this cluster here and slowly migrates over to the cluster on the left. And if you look at the pseudotime ordering, it's very representative of this ordering as well, letting us know that this is this makes biological sense, at least, or this is a decent ordering of the cells. And now it allows us to get these different trajectories of cells and or of genes, excuse me. And the next step is we want to learn dynamics on this, something like this discrete linear system. Because the thought process here is once we have dynamics, then it's just a control problem. Picking out which transcription factors and when to add them into the cell is just estimating a control law. And so this is the way that we approach this thinking. And what's left to do here, now that we have the gene ordering, is we want to build this linear system, and we need to figure out, what's the best coordinate frame. The dimensionality, obviously 20,000 genes is extremely large, and we don't have that many time points, so we need to find a good reduced representation of each cell, how many time steps we should use. So again, we have about 7,000 cells. So if we do something like binning, for example, or this polynomial fit, how many points do we want to take from either the polynomial or from like from a bin? And then the last thing is that we do know we have some information on the gene network, which genes interact with each other genes, and we can use that to drastically reduce the number of parameters within the A matrix, basically masking all the parameters that we know correspond to genes that won't interact with each other. And so how do you pick this sparsity pattern? There's different ways in literature, and so we're going to look at a variety of different graphs, such as like correlation, to see which one works the best for our methods. So now I'll end with some conclusions, which is basically just digital biology has the potential to revolutionize the medical field and biology because of all this rapid collection of data and this high dimensional data and the advances that we've seen in AI and machine learning. But there's still a lot of work to be done to fully reap these advancements from other fields. We've seen this with the Geneformer example, but there are a lot of ideas out there scaling up the Geneformer that might be very promising for improving these methods. And then lastly, breakthroughs in AI can be brought to biology to further accelerate discovery. Funding for this work is brought by DARPA and the Air Force. And I appreciate your attention. I'm happy to take any questions at this time.
No, thanks, Nick. That was really interesting. So first we'll take questions from people in the room. If anyone has any questions.
I had a few questions.
Okay.
Yeah, so when you showed that live cell imaging, I think it was in slide 22. Could you explain like how does it tie into the overall pipeline of the reprogramming? So does this serve as some sort of validation experiment when you actually try out some recipe for reprogramming, or how does it fit into the workflow?
So these are two transcription factors that we know a little bit about. So MyoD1, if you remember, was used to move a fibroblast to a myoblast cell.
Okay.
And then PRAX1 is a transcription factor that is highly active in fibroblasts, but not so much in the myoblast. And so we're looking, I guess, at the effect that these transcription factors have, or just sort of showing that they can influence the cell cycle dynamics. The idea would be using these two together in like a hybrid reprogramming, which is different from what Weintraub did in '89. But I guess if you, I guess it's mostly just showing like how cells can respond to these exogenous inputs, and how like adding PRAX1 is going to have them entering the cell cycle a lot more actively, which is sort of what we want for reprogramming, and how maybe that could improve the efficiency is by using these two transcription factors together instead of just MyoD1, where you don't see as much cellular activity.
And then one more question is that are there like other methods for performing the tokenization that you described at the beginning, where you are trying to get these to something more amenable for these natural language models? Because I think the tokenization is something that the outputs of foundation models are very sensitive to, and sometimes they can break due to bad tokenization. So is there some sort of prior on what a good tokenization should look like for these?
I think it's largely still an open question. There are other ways of doing it. For example, you can bin gene expression values. But then that runs into the issue of like, how do you choose the resolution of these bins? Other ways of tokenizing. I mean, yeah, I guess all I can think of right now is like binning for another way. I think what we'd want to be able to do is use like the full gene expression values somehow to add those, keep those within the tokens, because that does contain a lot of information, more so than just the ranking.
Yeah. Questions. Okay, I had a question. So you did mention AlphaFold, and I think there's AlphaFold 2 as well right now. So what are the potential challenges of protein folding? Is it the dimensionality, or like is it the sparsity of the data?
What are the challenges of protein
like, how is
folding?
it different than generating like images? Like, how does it change?
I mean, so like before AlphaFold, right, they would have to do the experiment, like do actual experiments to see how a protein would fold. How is it different from generating images? Can you?
Yeah, like what's the challenge there? Like, is it about the dimensionality? Like, what's the novelty that AlphaFold actually brought?
The novelty of AlphaFold. I think it was probably the way that it was able to incorporate different knowledge from physics and from like molecular biology and incorporate that into an actual neural network structure that could reliably produce good estimates of the folding.
Yeah, actually, one more question. I think maybe it was slide 28. So maybe it was like a previous slide, but I think where you were appending the TFs to the inputs.
Like 29.
Yeah. So all these TFs were pre-selected for these cases, right? You already had a knowledge about these TFs would result in a target cell, like, or they were, like you did mention towards the end that system ID, that's one way of selecting these TFs. But has there been like previous research on how you can select these TFs to induce desirable cellular properties or like desirable cellular?
So these transcription factors come from an algorithm that the lab has developed. It is actually, I think I'll use the appendix. So it comes from this dynamical model that we were able to put together. This was, I believe, in 2017, where we formed this time varying linear system, and then perform optimization over the inputs U to find transcription factors that will lead the state to a target state. So this is the dynamics here. AK is basically a rank 1 update of the identity matrix that satisfies this property here, where xk plus 1 should equal AK xk. And then we have a B matrix that was formed using our knowledge of where transcription factors bind on the genome, and using that as a proxy of which genes are going to be activated by what transcription factors. And then it's just doing some sort of like discrete optimization over which transcription factors should be added to the cell to move it towards the state that we're interested in.
Right. Yeah, thanks. Now, are there any other questions in the room? If not, like people on Zoom, either they can type it on chat or just unmute themselves if they have any questions.
Yes, so thank you, Nick, for that wonderful presentation. Very interesting. So my question has to do with benchmarking of these techniques. I wonder if you considered using some of the data coming from some large screening libraries, so like George Church Lab at Harvard. In 2021, they had a paper, I think in Nature Biotech, where they profiled a library of about 1,500 transcription factors to see their impact on HSPC reprogramming. So in that data, they have both like positive and negative hits. And I wonder if you're considering doing some benchmarking with that kind of data that is experimental.
Yeah, I think we do want to incorporate data from these perturbation screens, because that's going to tell us how like perturbing a cell is going to have like the downstream effects of perturbing a cell. And that's something that is not really contained within our cell cycle data, because that's the nominal cell cycle. As far as like benchmarking, like the foundation model, I think it is difficult. I guess what you're saying is like try to doing the same perturbation that is contained within the data set, and then look to see on the actual data compared to the Geneformer embedding. Yeah, I think that's something that we could look at. Again, like from the Geneformer, we will really only get a direction. We can still compare directions, but they're it's only like the first step, I guess, of the effect of the perturbation, whereas like these perturbation screens have the cell reaching some sort of like, state that after like a period of time where like it's actually had time to like transition or perturb. So there's a little bit of a challenge there in comparing them directly, but it would definitely be something to look into. For now, what we've done in terms of benchmarking is just taking like known transcription factor recipes that we know will take one cell to another cell, and seeing if the results match what we've found experimentally and in literature.
Mm-hmm. But with that, do you need like some control negative perturbations, which could be like randomly sampled genes to see how sort of the reprogramming fate is very sensitive to like the combination of genes. So you could like try different combinations, or try this sort of like you can drop one of the transcription factors in the set that you use and see how that affects the outcome.
Oh, you're suggesting look at like the sensitivity.
Yeah, like the sensitivity. Yeah.
Yeah, that would be interesting. And then in terms of also these like perturbation screens, we've also been looking into different neural networks that have used the perturbation screens to train and to predict like the effect of different transcription factors and different perturbations on cells. And so that's also like another avenue that we're exploring, is trying to use these networks that are trained directly on perturbation data, and it should have a better understanding or knowledge of how gene perturbations are going to affect other genes within a cell, using those as a way to look at you know, different transcription factors that might be useful within like dynamics, for instance. Something like a almost like a B matrix here, where you'd look at, you know, what is the perturbation and then how does that affect different genes.
That's wonderful. Thank you.
Thank you.
Are no questions? Yeah, let's just thank the speaker again.
Thank you, guys.